Welcome back to botany class. Let's look at glance of today's learning objectives. Now we are discussing about transgenic plants. Let's continue from virus resistance, FlavrSavr tomato, golden rice, that is biofortification, polyhydroxybutyrate, polylactic acid, green fluorescent protein, bioforming and its limitations. One more, bioremediation. These are the learning objectives. Let's begin from virus resistant. In the previous class, we discussed about transgenic plants. There are number of transgenic plants. Let's start from virus resistance. You know there are number of virus which attack the plant crops. Due to this, it affects the yield. Even it leads to death of the plant. So that disease resistant varieties, virus resistant varieties are developed using genetic engineering technology. So one of the genetic engineering technology is to produce virus resistance. What's this virus resistance? Viruses which attack plants. When it attacks the plants, it affects the yield. So economic value of this product less. So the plants are modified by introducing some genes. Previously you learned about this topic, transgenic plants, how it occurs. Because of the biotechnology, it produces recombinant DNA or DNA, recombinant DNA, introducing a desirable gene which produces the enzyme which deactivate the viral DNA. So if virus resistance, the plant, transgenic plants, when it develops as virus resistant, if the virus attacks the plant cannot be affected. Such a way the plant can be modified. That's called virus resistance. So how it works? This rDNA, already the plant gene is modified by resist to virus. So this gene, this new gene which secrete enzymes which degrade the or inactivate the virus DNA. When the virus DNA inactivate, it do not cause any disease. So this method of transgenic plant is called as virus resistance. Then let's see some more examples of this FlavrSavr tomato. So this is one of the tomato plant, FlavrSavr tomato. Tomato and so many fruits, very easily it gets spoiled. When it is transporting long distances, it gets spoiled. So that there is a bacteria, normally in the plant transgenic plants are developed by using Agrobacterium tumefaciens. So this bacteria is very much useful to produce or make modified gene or transgenic plants. So this tomato variety is modification of the Agrobacterium mediated variety. Agrobacterium mediated genetic engineering technology can be used and develop this FlavrSavr tomato. What's the functioning of this? It delays the process of ripening. The ripening process can be delayed. So it restores the color of the fruit and the flavor of the fruit. So the fruit color, tomato fruit color, and it do not get spoiled. So it is in solid state. Few more days. So it delays the process of ripening and delays rotting. So for rotting this, delays the process of this they are introducing a gene, that is the antisense gene. This is the gene, antisense gene. Introducing, when it is making this agrobacterium used as a vector, the gene introduced through agrobacterium is antisense gene. So, this antisense genes prevents or interfere, that means it prevents the production of the enzyme polygalactonase. You remember all these enzymes. Here you are going to learn number of enzymes and number of examples. Here the gene which is introduced for making this tomato is antisense gene. The name of the gene is antisense. What's the function? It interferes the production of polygalactonase. So, when it affects or prevents the production of this enzyme, what's the name of the enzyme? Polygalactonase. Polygalactonase enzyme, it prevents to produce this enzyme. Antisense gene prevents for the production of polygalactonase enzyme, so that it delays the process ripening. So, it can be transported very long distances in a solid state. Such a way that this tomato is modified as a transgenic. Another example, golden rice. Golden rice. Golden rice, you know what's the binomial of rice? It's Oryza sativa. Oryza sativa. Golden rice. The transgenic form of rice or the modified form of rice is named as golden rice, which is biofortification rice. Biofortification. What's this biofortification? Fortification means the nutrient content can be added. Extra nutrient can be added. You know, in rice plant or Oryza sativa, it is rich in starch. So, which is the staple food of our area. So, this Oryza sativa, this rice which contains starch is abundant in this. In addition to this, some extra nutrients are added. That type of transgenic or that type of modification and produce a rice called golden rice. Here what is introduced? In addition to starch, there are beta carotene is added. So, carotene content is added. So, when you consume this rice, golden rice, which produces starch, in addition to starch, it also gives carotene. So, beta carotene gene is introduced in it, which is developed by Ingo Potricus and his group. They develop such a transgenic rice is called golden rice. That name of this rice is, the process is biofortification. In addition to the normal endosperm of the rice, there are 3 genes are introduced in it. The 3 different types of genes which are derived from carotene producing beta carotene, which is derived from daffodil plant, and one is derived from soil bacteria and rice endosperm. So, 3 different types of genes can be introduced. The name of the gene is PSY. The first type of gene is PSY. Another one CRT1 and LYC. What are these genes? 3 more question which is very important. The 3 different genes introduced into golden rice for biofortification is PSY. The first type of gene is called as PSY, that's phytoene synthase. The name of the gene is phytoene synthase, which is derived from daffodil. The binomial of daffodil is narcissus plant. So, it is derived from narcissus, this gene, PSY gene. What's this PSY? Phytoene synthase gene. Second type of gene is CRT1. That's derived from one of the soil bacteria. So this is derived from one plant, daffodil plant, phytoin synthase enzyme, commonly called as PSY. Another one, CRT1, derived from a soil bacterium, that is Erwinia aridorora. So this is bacterium, soil bacterium. Third type of gene is LYC, lycopene cyclase. Lycopene cyclase. This is derived from rice endosperm. The normal rice endosperm which stores starch. This golden rice which contains, in addition to starch, carotene, beta carotene. So when one consumes this golden rice, they are getting carotene in addition to starch. Why they develop this? To control xerophthalmia, that is child blindness. So many people, many children, they are affected. So it contains this gene, the beta carotene, which encodes vitamin A. To avoid this deficiency or to control this xerophthalmia, the plant can be modified. Oryza plant, Oryza sativa, or rice is fortified. The extra nutrient can be added. And there are 3 different genes. Here 3 mark question, very important. The 3 different genes present in this golden rice is PSY, CRT1, LYC. What is this PSY? Phytoin synthase, derived from daffodil. This CRT1 derived from a soil bacterium, Erwinia aridorora, and LYC is called as lycopene cyclase. Lycopene cyclase, which derived from rice endosperm, which is developed by Ingo Porticus and his group. This is the question. So first question, virus resistant. What is virus resistant? 2 mark question. Or sometimes they may ask for 3 mark question. Then FlavrSavr tomato. Write about FlavrSavr tomato. It's also 3 mark. Then golden rice, 3 mark question. And this genes, important. Then what are the here this golden rice and biofortification. What's the use of this? Or these all plants are transgenic plants, or they are called as GM plants. GM. What's GM? Genetically modified plants. So what are the benefits of these modifications? These are first 3 examples you learn: virus resistant, FlavrSavr tomato, and golden rice. What's the use of this modification? Why we are modifying? Is there any benefits when the genes are modified or when the plants are modified? It produces, what's the benefit of this? It produces high yield. The ultimate aim of plant breeders are to increase the content qualitatively as well as quantitatively. The content or the products can be increased. The yield must be increased. So when the plants are modified in such a way, it increases the yield, high yield. Also, when we are applying herbicide or pesticide without modifications of these plants, if it is not transgenic or genetically modified, we can use pesticide, herbicide, insecticide, all kinds of chemicals must be used. It should be avoided. When the plants are modified in such a way, it should be avoided. So pesticides, spraying pesticides, avoid. There are 70% reduction of pesticide usage. So chemical usage, chemicals when the chemicals are used in the form of pesticide or herbicide, it not only affects the weed or the pest, it also affects the crop plants yield. So little by little it enter into the body of living organisms. To avoid this, 70% reduction in the usage of pesticides. That's one of the benefit. And when we are using this herbicide or insecticide, that's chemical substances, it also affects the soil environment. So this conserves the soil environment as well as the microbes present in the soil. So these all crops we are planted in the field, in the soil. So this chemical substances, when we are spraying some substances in the form of chemicals, it not only affects the weeds or pests, it definitely affects the soil, water and the surroundings. So it can be conserved. So conservation of microbial population and conservation of soil also possible. So these are the benefits. So benefits of genetically modified plant, 3 mark question. Next 3 mark question is, what are the benefits of genetically modified plants? Then, though there are some benefits, there are some risks also. By modifying this plant or due to transgenic plant, there are some risks. What are the risks of this? It affects liver, kidney, the functioning of liver and kidney. Also it causes cancer. So it causes some diseases because of modifying this, because we are changing the gene or genome. So it causes disease, it affects certain organs, and it affects due to radiation, it causes, due to mutation, it causes cancer also. Then hormonal imbalances. The nature can be altered through this method, this rDNA technology or genetically modifying technology. The normal or natural nature can be modified so that it leads hormonal imbalances, and it due to this reaction it produces allergies also. So certain times nucleic acid content, due to this alteration of this, it causes allergy reactions. That is the limitations or the risks of this genetically modified plants. Then let us see the next topic, polyhydroxybutyrate. So this microbial, this plants, not only the plants alone modified, there are some microbes can be modified. So here polyhydroxybutyrate. So polyhydroxybutyrate is a synthetic polymer. So synthetic polymer is non-degradable polymer. The polymer is non-degradable. The non-degradable polymer can be changed by genetically modified to degradable polymer. For using this degradable, this polyhydroxybutyrate, PHB, it's a synthetic polymer. So by making this as a degradable polymer, there are 2 genes are used. One is PHA and PHB. PHA is polyhydroxyalkanoates. Polyhydroxyalkanoates and polyhydroxybutyrate. So this produces this synthetic polymer which is non-degradable, which develops, which polluted the soil, as well as it produces, when it burns, it gives this dioxin. So this dioxin causes cancer. So that it's made as a degradable polymer. So degradable polymer, this polyhydroxybutyrate, PHB, the butyrate, polyhydroxyalkanoates and polyhydroxybutyrate are the degradable. It changed the synthetic polymer into synthetic non-degradable polymers into degradable polymers by using this. So this one, what's the use of this degradable plastics or this polymers? So in the medical field, it's very much useful to producing the drugs, the drug delivery. There are so many packages and all made up of non-degradable substances. It can be biodegradable substance. So it's used in drug delivery and scaffold. Scaffold, there are some parts or tissues which is made like heart valves or some tissues can be made in biodegradable substances. So, polyhydroxybutane. In the same way, polylactic acid. Polylactic acid is also a thermoplastic. So, this polylactic acid produced is derived from the common substances like cornstarch and cassava, the root that is Manicata esculenta. From Manicata esculenta and cornstarch, this polylactic acid is obtained. The thermoplastic can be obtained from this common roots, the cornstarch, starch and Manicata roots, Manicata esculenta roots. From this it is derived polylactic acid. Another question is green fluorescent protein. Another type of protein is called as green fluorescent protein. This protein is named as green fluorescent protein, commonly which is called as GFP. GFP, green fluorescent protein, green fluorescent protein. What's this green fluorescent protein? So, this is a type of protein which contains 238 amino acid residues. It is a kind of protein, green fluorescent protein contains 238 amino acid residues of 26.9 kilodalton. So, this much bioresidues are present in this 238 amino acid residues. Amino acid residues 238, the weight is 26.9 kilodalton. So, it is it exhibits a green fluorescent light when it is exposed to ultraviolet blue spectrum of ultraviolet. It gives or exhibit green fluorescent light. What's the use of this? This is derived from, this protein is derived from jellyfish. Jellyfish, the name of the jellyfish is, scientific name is Achoria victoria. Achoria victoria, jellyfish. So, this produces a chromophore. What's the use of this green fluorescent protein? It's the expression of reporter, to report something, or it's used as a biosensors. For noting this or this expression to find out some substances, this is used as a reporter expression. To express this green fluorescent protein is used. Also, it used as a biosensor to indicate, it used as a biosensors. Next question, biofarming. Biofarming is otherwise called as molecular farming. What's this biofarming? It's a process by which it's used in pharmaceuticals. So, biofarming is used in pharmaceuticals. So many medicinal substances are made by this biofarming. What are the limitations of this? The limitations of biofarming is this biodegradable substances. Biodegradable contaminants can be transformed. Contaminants, biodegradable contaminants, that's the pollutants, can be transformed as a biodegradable. The contaminants are transformed into biodegradable substances. And the condition of this all biodegradable substances, or this process biodegradable, which is used for the area where the contaminated is present, the site of contaminated. It's the conditions at the site of contaminants. Where the contaminants are there, the pollutants are there, that site can be chosen, and the condition is to degrade as a that is eco-friendly approach. By eco-friendly approach, without making any hazard to the soil or water or the air without polluting anything, it can be make it as a biodegradable substances. Before using these all technologies, this biodegradable, the microbes can be modified. Here for using this process, microbes can be used as a transgenic or genetically modified. So, at first, before going into the process, there is a test, small scale test, that is small scale test must be performed, because some substances which emits different kinds of chemicals and it also polluted the air or water or the land. To avoid this, a small scale test must be performed. Then the genetic engineering, for all these techniques, the technology involved in this is genetic engineering technology. So, this genetic engineering technology produces a modified microorganisms. These all microorganism, bacteria, is in the form of bacteria or plants or any other animals. This all are modified. The microorganisms are modified microorganisms. So, the nature can be altered and it produces a new modified organisms can be produced. So, these are the limitations of this. So, limitations, 3-mark question or 2-mark question, sometimes 5-mark also they may ask, all kinds of limitations. This point wise question important. So, you learn all this for question oriented, the exam oriented, prepare it well. Then there are some strategies to bioremediation. What are the strategies of bioremediation? So, next topic you are going to learn is bioremediation. What's meant by bioremediation? So, remediation, biological remediation. The biological remediation, here the contaminants can be make it as degradable substances. So, there are some process, some examples of bioremediation. What are the examples? Phytoremediation. So, first example is phytoremediation, then mycoremediation, bioventing, bioleaching, bioaugmentation, composting, rhizofiltration and rhizostimulations. Here rhizostimulations. So, bioremediation, biological remediation. So, for this they are using green plants. You know, normally we are making plants around our houses, or if any buildings are there, around this there are some green plants. Knowing or unknowingly, these plants we are planting. What's the reason? These plants are observing this, during respiration it takes oxygen and releases carbon dioxide, and the carbon dioxide can be taken during photosynthesis and it evolved oxygen. So, oxygen is one of the air purifier. So, when the oxygen content is released, this oxygen is a fresh air. So, the carbon dioxide can be utilized by the green plants. So, everywhere the green plants are utilized. Green plants are very much useful. That is one method. So, green plants are used for bioremediation to absorb the unwanted gas and releases the pure oxygen, the pure gas. So, phytoremediation, phyto means plants. So, remediation, by using this is the process by which the green plants are used. Because of this reason, because of photosynthesis, by photosynthesis process, for photosynthesis process, plant absorb carbon dioxide and release out oxygen. So, that is one of the phytoremediation. Likewise, mycoremediation. What's meant by myco? Myco is fungus. Fungus. How fungus are helpful? Fungus are non-green plants. Other green plants, that is phytoremediation. Green, because of the presence of green, it produces this oxygen, because it evolves oxygen during photosynthesis. How this fungus are involved? in bioremediation, fungus, which is very much useful by degrading the substances. The complex substances can be degraded by in the form of the complex substances or degrade into smaller substances by means of fungi as well as bacteria. So, saprophytic fungus are very much useful. So, mycoremediation. So, degrading substances which degrade the substances and it enrich the soil fertility. Then bioventing, another process by bioremediation is bioventing. That is the process by which increase the oxygen content. So, it can be increase the oxygen content. Then bio-leaching. Bio-leaching, it recovers the metal pollutants. The metal pollutants can be recovered by this method called bio-leaching. Then bio-augmentation. This is bio-augmentation by adding some microbes, addition of some microbes into the soil to speed up the process. What process? This recover metal pollutants can be absorbed or it can be degraded. The degradable process can be speed up by involving, adding some microbes. If what are the contents, the contaminants are there, depending upon the contaminants, what are the microbes are in need for that area which can be added. If fungus needed, they add the fungus. If bacteria is needed, they add the bacteria to speed up the process, speed up to degrade the substances. Here mainly it is used for the managing the wastewaters, or the soil solid wastes. Next process, composting. Composting is this compost, the solid materials, solid substances can be degraded into or compost and it make the soil fertility, enrich the soil fertility. And rhizofiltration. Rhizosphere, here rhizofiltration, it's the root and the soil, that area covers the area, it degrade the organic compounds and it uptake the metals. During this process, rhizofiltration, it uptake all the metals and it degrade all the organic compounds. Organic compounds can be degraded. Rhizofiltration, the usable substance the plant can take by means of its root, it absorbs. Rhizostimulation, this is reduction in the toxic materials. The toxic materials can be reduced. Due to this rhizostimulation, it's the process by which stimulate the process of degrading to reduce the toxic substances. So, if waste materials are there, it's in the form of liquid or solid, the waste which contains some kinds of toxic substances. This is the process rhizostimulation to reduce the toxic substances. Then, so this is bioremediation. Bioremediation. The limitations of bioremediations are this. Because of biodegradable, what's the use? What's the limitations of biodegradation? Bioremediation. So, bioremediation is the process by which the contaminated can be degradable. Degrade. So, it's degrade because the contaminants are transformed. All these contaminants, the pollutants can be transformed. So, these all pollutants finally it mix with the soil and the plants can utilize this. And there are some limitations. What are the limitations? This biodegradable contaminants are transformed and conditions at contaminated site. Small scale test can be performed before that, and genetic engineering, the modified microorganism. All this process make a modified microorganisms. One more question. Here are some examples of polyhydroxy alkanoids. This polyhydroxy alkanoids, this gene can be produces this polymers, degradable polymers or made by use of some microorganisms. There are some bacteria, gram positive bacteria and gram negative bacteria. This PHA can be obtained from this bacteria. The gram positive bacteria, example Bacillus megaterium and Bacillus subtilis. Bacillus megaterium and Bacillus subtilis, and Coryn bacterium glutamicum. These three examples are gram positive bacteria, and gram negative bacteria Pseudomonas species and alkali genes eutrophus. Pseudomonas species and alkali genes eutrophus are the examples of gram negative. So, let us see once again what are the topics you covered today. First, virus resistance, then write about FlavrSavr tomato, then learn the question golden rice. Note all this: golden rice, here 3 genes, biofortification, biofortification example golden rice, then polyhydroxybutyrate and polylactic acid, then green fluorescent protein, bioforming, then bioremediation, and these are the examples for polyhydroxyalkanase PHA obtained from gram positive and gram negative bacteria. Finally, write the limitations. So, today this all question you will learn well, and next week you have the test. Prepare well and write the test. Thank you.